All right. All right. In the Father, Son, Holy Spirit, amen. God, grant me serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. The Father, Son, Holy Spirit, amen. My apologies, I got going a little late this morning, so let's start with, see if you all have any questions. Questions about fatty acids, questions about carbohydrates, questions, confusion. No, we will move, okay. So phospholipids. Oh, two properties of all lipids. Two properties of all lipids. Okay, John. So they're insoluble in water. What's the other one? They have lots and lots of what? Okay, I'm going to get this. Emily. Lots of hydrogen carbon bonds. Lots of what kind of covalent bonds? Non-polar, right? I'm not going to go over electronegativity again because we're drilling that in. Okay, good. So we talked about triglycerides and phospholipids. They have something in common to them. What are some things that, what's something that both of them have? Let's go with Kristen. Kristen? Don't know? Okay. So they both have a, what's this molecule? Oh, glycerol. They both have a glycerol backbone. And they both have fatty acids attached to that glycerol backbone. So what's one of the main differences structurally between triglyceride and a phospholipid? Yeah. Okay, phospholipid have phosphate group. What's bound to this glycerol? Anthony. And what's the clue that the triglyceride has 3 bound to it? Triglyceride. 3 bound to glycerol. Good. Okay. So why don't they call this a diglyceride? Well, because they call it a phospholipid. Get over it, Kyle. Get over it. I'm just kidding. Okay, so anyways. So you need to know how to draw this thing in the cartoon form. Fatty acid, glycerol, phosphate, and then this choline molecule. So we ended here, and we identified then that this molecule has 2 regions that have very different chemical properties. So let's start with the property that we've talked about already today. Oh, wait a minute. Something happened. Sorry, I got to go back to Zoomville. Let's see here. Oh, they're all there. Okay. All right, let's move you guys off the screen. I'm gonna minimize you guys. Hopefully that'll show up if someone has a question. Okay. Let's start with what we know. These are fatty acids. So what property are these fatty acids going to have? Jonah. Nonpolar. Nonpolar means, how do they interact with water? Marie. So we call that hydrophobic. Good. So the one region with the tails, these are called tails, by the way. Capes. Superhero, no capes, right? Incredibles, no capes. Of course, maybe we could say these are legs, whatever. So we call these the tails. So these are hydrophobic tails. And then, what do we notice about, so this is very nonpolar. What do we notice about this part up here? What do we see? What do we see? Yeah. It's not only is it highly polar, it's charged. So, what just happened there? Go away. So it's charged. That was weird. Good. Oh, I gotta do my first advance with. Okay, so it's charged. So that means that this molecule has 2 different properties.
I'm sorry, Father Sean.
Yo.
Change the camera angle back there to show the board, if possible.
You don't see the screen?
No, I'm looking at the corner of the room, sorry.
Okay, so let's try to share the screen. Let's try this again. How about now?
Oh yeah, I'm seeing it now, but it's in terms of the classroom, like if you're pointing stuff out on the board or making notions in person, I can't see them.
But you're not seeing the screen?
No, I'm seeing the shared screen perfectly fine. It's your actual camera in the back of the classroom.
Should I turn it off?
No. If you could just like angle it to the right a little.
I can't do that.
Okay. It's fine then, thank you.
I don't think I can change that. Oh, is it over to the corner or something?
Yeah.
Somebody came in and must have done that over the weekend. I don't know.
Yeah.
Sorry about that.
All right, thank you.
It's like NBC, NBC Studios, ESPN. It's always something different. I can see somebody must have knocked it over or something. It looks like it's all torqued. All right, let me try this.
That's good.
Better?
Much better. Thank you.
Got it. Okay. I freak out when anything's wrong, so I just froze. Good. Thanks. Thanks for letting me know. Okay. Where were we? Oh, amphipathic. So amphi means a couple of things, multiple things, like amphibian, can live in water, live on land. So they're amphipathic, which means they have a non-polar region and a polar region. So we call the non-polar regions the tails, the polar region the head. And if it's drawn, oftentimes you'll see it represented like this. So what then, since this has a different structure, we know that in cells, this is going to have a different function. So let's take a look and see what the function of triglycerides, how they function in water, and how phospholipids function in water. So if you were to take a cup full of triglycerides and just sort of pour them in, you would get these little droplets. Kind of like if you had oil in water, right? Little droplets of oil being suspended in water. The heads on these things are it's a very small region, right? Because they are called fatty acids. So there's some hydrophilic region, but it's not very prominent. But because of the structure of the phospholipid, if you were to take a cup of those and just sort of pour them in a glass, you would actually, if we could look at it with a strong enough microscope, you would actually see that there would be formed in this glass or in this beaker a bilayer of these phospholipids. So what happens is that the tails, what kind of interaction are the tails going to be having? Not with the water, but with themselves. So think about that for a moment. What kind of interaction will the tails be having? Let's go to, I see a hand over there. How about Ashley? Very non-polar. If we put a bunch of non-polar R groups from an amino acid, when they came together, we would call that a, Donovan? Oh, I thought you raised your hand. Okay, I saw your hand go up. Hydrophobic exclusion. So what happens then is that these nonpolar tails, they get drawn to one another. So they're facing each other. Hydrophobic exclusion, they push the water out. And then the polar heads, because they are hydrophilic, they then interact with the water. And phospholipids then comprise the major component of plasma membranes. And I'm going to say this now, and we'll come back to this in a moment or later. But one of the major benefits of this structure, phospholipid structure, is that the cell does not have to exert any energy to hold the membrane together. They just do it, just by the property of those phospholipids. So once the phospholipid is made, shuttled out to the plasma membrane where it's supposed to go, it just goes in, and it's there. So it's awesome. All right, steroids. Steroids. Every time I think of steroid, I think of, Arnold, I said Arnold Palmer, Arnold Schwarzenegger. You don't even know who knows who that is? All right, Terminator, good. So, steroids are also lipids. They have this basic backbone structure. You need to be able to identify this. Identify this. Know it. This structure right here. Know it, know it, know it, know it, know it. This backbone is these are all hydrocarbons. So this backbone is very, very hydrophobic. Don't be fooled by the functional groups that you might see on this ring, on this backbone structure. All right? Okay, this is cholesterol. So cholesterol is the basis of all of your steroids. That's what your body uses. So people tell you that cholesterol is bad for you. Well, yeah, a lot of cholesterol is bad. But if you don't have any cholesterol, it's hard for you to actually make steroids. Steroids. And what makes it a steroid? This backbone structure. So, hydrocortisone, otherwise known as cortisol. Who's heard of that before? Okay, so when we think of cortisol, what do we think of, Kate? It is a steroid. And we've heard of maybe studies are in the news. It's released during when you're experiencing what? Stress. So it's a stress hormone. And we notice that we've got all kinds of polar functional groups, like this one here. What is that? Right there. Carbon double bonded with an oxygen. That's a carbonyl. How about here? We'll go to the board. Eden. Right here. Boom. What is that? Hydroxyl. Okay, good. So don't be fooled by these polar side groups. This is a non-polar molecule. There's testosterone, or what's called sex hormone. All right. But the main point of this is that this backbone is going to be the same in all steroids. It's going to be the same in all steroid hormones. And all of these are hydrophobic, hydrophilic. This is going to play a really important role when we start talking about how the body communicates with different parts of the body. Because when we use steroid hormones, because they're lipophilic, because they're hydrophobic, they can go right through the plasma membrane, right through the cell membrane into the interior of the cell. Yep, there's Gollum again. Thank you, Gollum. So, functions. Cholesterol has a different kind of function. It is the basis for synthesizing all steroids, but it also modifies the properties of cell membranes. Cortisol and testosterone, these function as hormones, molecules that are released into your blood, that circulate around your body and have an effect, some kind of effect. Okay. So, table, lipids. I guess, see, I don't even know if I want to fill this table in with lipids because it kind of breaks the rules. But fatty acids, I guess perhaps if we're talking about either triglycerides or phospholipids, we could say the fatty acids are the monomer. I'm not going to worry about the bond. But you should definitely be able to, by looking at the structure or the kind of molecule, the kind of lipid that it is, you should know what function it plays. Should know what function it plays. Fatty acids are used for energy. Triglycerides are used to store energy. Phospholipids are used to build membranes. Steroids, for the most part, are hormones. All right, 4 functions, different kinds of molecules, but they're all lipids. Okay, questions about lipids? Questions about lipids in TV land? Can you still hear me?
Yep. I can hear you.
Okay, just making sure. All right.
Gotcha.
Okay, cool. All right. Let's talk about the types cell parts. You were to have completed the cell part table. We are going to go through it today. Whatever we don't get through, I'm posting online, and you can complete that because we're not gonna move on. So the table has the actual part, or part is kind of like, I guess, but in general, when we talk about cell parts, what's a more technical term for that? Organelles, okay? I guess cell membrane is not, I guess it is kind of an organelle, right? There's going to be structure, like what the structure is, what's the function of that structure, and then the question is this found in prokaryotic cells, eukaryotic cells, or both? So I'm going to ask you guys a quick question. What in the heck distinguishes a prokaryotic cell from a eukaryotic cell? What's the main thing? Okay, so we're going to see when we go back over this distinction between prokaryotic and eukaryotic cells that eukaryotic cells have a true nucleus. The word itself actually means true nucleus. All right. Sir. By the way, did you guys bring, you just brought the table, it's fun. All right. So, membranes. Membranes. Who can tell me what can we say about membranes? I guess it'd be kind of hard with the mask on, so I'll just yell at you guys when it comes to some longer things. Phospholipid bilayer, right? That's the main thing. There are proteins that are in the membrane. We will definitely talk about this. What's the main function? Main function of the membrane. If you were to sort of give me the main function of the membrane. What's the main function of the membrane? Christina? Okay, so it's forming a boundary between the outside of the cell and the inside of the cell. And it also contains they're mainly proteins, which allow signals to pass from the outside to the inside or the inside to the outside. Good. Okay. Plasma membranes, are they found in prokaryotic cells, eukaryotic cells, or both? Ashley? Both. So all cells have plasma membranes. Cytoplasm and cytoskeleton. Let's start with cytoplasm. What is the cytoplasm? It's got lots of names. Donovan. Okay, so that's cytoplasm. We also call it cytosol. It's got lots of different names. So did everyone hear that? It's got like all the stuff that the cell needs for life, basically, is in the cytoplasm. And it's more of a gelatin. Okay, that's a good distinction. So it's not fluid inside. Okay, even though we talk about intracellular fluid. It's not like the cell is filled with water. Okay, although there's a lot of water there. Cytoplasm, is that found in eukaryotic, prokaryotic cells, or both? Yes, Jordan. Jared. By the way, Jordan's right. Justin. Jordan. Yes, both. So all cells, if it's a cell, it has cytoplasm. Beautiful. What about cyto? When you hear the word cyto, by the way, cyto, that means cell. So cytoskeleton. Cytoskeleton, what is the function of the cytoskeleton? First of all, what is it? Got anybody? Anybody? Okay, Jonah? Okay, microtubules, microfilaments, and what kind of molecule are they? Starts with a P, ends with an N. Proteins. So we've got these networks of proteins that help maintain the shape of the cell. You can even think of it as being like a scaffold. So for example, the fence that's been put up, the chain link fence, those bars, you could sort of think of that as like being the plasma membrane of that fence. And never mind. That's a bad analogy. Why did you even suggest that? Sophia's making fun of you. Just to let you know that. Anyways, okay. Prokaryotic, eukaryotic, or both? You, both. John, you said both. Cytoskeleton. Anyone? Okay. Okay. In general, what supports the cell in a prokaryotic cell? In general, big picture here. I heard something. Remember? Okay. So, cytoskeleton, we're going to say mostly eukaryotic cells, some prokaryotic cells have it. If you're trying to distinguish whether a cell is prokaryotic or eukaryotic cell, don't go looking for the cytoskeleton. That's a little dicey situation. Okay. Region of the DNA. So we get some important structures here. Nucleus, right? Nucleus is the largest organelle. What does it contain? What's it contain? DNA. DNA. Yes, DNA. What's the bond that holds? So what are the monomers?
Nucleotides.
Nucleic acids. What are the bonds that hold those together? Phosphodiester bonds. Okay. Anyways, good. Nucleus. Around the nucleus is what's called a double membrane called the nuclear envelope. So the nuclear envelope is actually a double membrane. So really protecting the nucleus. So that's another structure. So pro-eukaryotic or both? Pro-eu both. John? Okay, so they both have DNA. Do they both have a nucleus? Okay, that's okay. They both have, you said they both have DNA. That's correct. Which cells have a nucleus? Let's go with Sam over there. Eukaryotic. Eukaryotic cells have a nucleus. That's one of the main things that distinguishes them as a eukaryotic cell. In prokaryotic cells, what's the structure? So in prokaryotic cells, they have DNA, as John said. It's sort of this loose sort of collection of DNA that's sort of held together with some proteins. What's that called? Marie. The nucleoid. Oid. So cells either have a nucleoid or a nucleus. Oid is prokaryotic. Us, what's not about you, it's about us. But oid. So DNA is either in a nucleoid or a nucleus. And that is important because there's another structure called, what's another someone come up with anything else? The nucleolus. Most people confuse nucleoid and nucleolus because they say, well, if it doesn't have a nucleus, it must have a nucleolus. No, if it has a nucleus, it also has a nucleolus. So look for the us. If you're trying to distinguish if it's prokaryotic or eukaryotic, if it's in you, it's got to be about us. So if you don't see us, it's not in you. Got it? Nucleolus, nucleus. You and us. It's easy. What is a nucleolus? Let's go to Sarah. Sarah in the back. Sarah, nucleolus. Okay. Inside the nucleus makes ribosomal RNA. Boom. Awesome. Chromosomes. Oh, so that's the function. Oh, prokaryotic, eukaryotic are both. We already said that nucleolus. If you're trying to figure out if it's the U, look for the us, right? So it has an us, so that means it's in eukaryotic cells. What's the function of the nucleoid is also we'll go back to nucleoid, is also information storage, right? Information storage. And finally, chromosomes. Chromosomes. What are chromosomes? Abigail, chromosomes. You got it? Yes. So it's how DNA is organized. So, chromosomes, eukaryotic, prokaryotic, or both? Okay, yep, Quattro. Both. Yes, that's correct. So in both prokaryotic and eukaryotic cells, so in the nucleus, the DNA is organized into chromosomes, and in the nucleoid, DNA is also organized into chromosomes. Good. Ribosomes. 2 subunits made up of RNA and protein. 2 subunits. Ribosomes are 2 subunits made up of RNA and protein. Eukaryotic cells, they can be structurally different depending on whether it's bound to the rough ER or out in the cytosol. What's the main job of the ribosomes? Cameron. Protein synthesis. Specifically, is that translation or transcription? Translation. Okay. We're not going to get into that too much, but we will talk about it a little bit, the difference between transcription and translation. What kind of cells have it? Eukaryotic, prokaryotic, both? Christina? Both, right? Cells got to make protein. Don't have RNA, you're not making any protein. You're not making any protein, what happens, Sophia? Go all the way. Dead, right? There you go. You can say that, death, cell death. Sorry. Okay. All right. Cell wall. Cell wall. Some cells have walls, some cells don't have walls. It's a rigid outermost layer of a cell. It's outside the plasma membrane. So first of all, let's ask the question. I just gave it away. What kind of cells have cell walls? Eukaryotes, prokaryotes, or both? Let's go, Landon. No. Oh wait, Tristan. Okay. So both eukaryotic and prokaryotic cells have cell walls. All prokaryotic cells have walls, I do believe. Prokaryotic, no, I do believe that, but don't quote me on that. There's a structural difference. So eukaryotic cells are made of carbohydrates. Prokaryotic cells are comprised of something called peptidoglycan. You can get that when I post it. Peptido, when you hear that, what should I think of? Peptide, peptido. Well, I heard something. Emily, yep. Did you say protein? Protein, good. And then glycan, like glycerol. There's a little bit of, yep, hydrocarbon, yep, peptidoglycan. Mostly more protein than carbs, okay? Good. Function protects the cell and gives the cell shape. So there's difference. Carbohydrates, eukaryotic cell walls are made of carbohydrates. And then prokaryotic cell walls are made of peptidoglycans. All right. Glycocalyx. What is this thing? The cell coat. What does this do? It's a layer on the outside produced by bacteria primarily, but some animal cells can make a glycocalyx. It protects the plasma membrane. And it's primarily going to be found in prokaryotic cells. Pili, not a pillow, but a pili. You can tell me what pili are. Pili as opposed to cilia. Cilia are different. Pili. Sophia. Okay, so hair-like structures. That's correct. What's the function? It adheres to other cells. And this is a way. Oh, and what kind of cells have these? So prokaryotic cells. So prokaryotic cells have pili. They're hair-like extensions of the cytoplasm. No, the plasma membrane. Their job, they can adhere to one another. And this is also how they exchange genetic information. So, if a bacteria, for example, becomes resistant to an antibiotic in the hospital, so you don't kill them all, you kill all but like a small group. Those begin to multiply. They come into contact with other bacteria of the same genus and species. They exchange genetic information. Now, all those other bacteria, they can now become resistant to the antibiotic. That's one of the ways that happens. Okay. Central vacuole. Well, let's skip that. Those are plants. Mitochondria, we know what that is, right? Powerhouse of the cell. Double membrane, tube shaped, looks like a kidney bean. What kind of cells have those? Pro, or both? Eukaryotic cells. Only eukaryotic cells have mitochondria. When you see large membrane-bound organelles, which we're going to hit a couple more here, those are going to be found in eukaryotic cells. That's one of the distinguishing characteristics of a eukaryotic cell. The reason it doesn't have a nucleus is because a nucleus is bound by a membrane. Sweet. So let's hit this endomembrane system. ER, rough and smooth. ER, rough ER. That's a membrane that has, what makes it rough? Ribosomes attached to it. Yes. And then the smooth ER is a membrane-bound structure that doesn't have ribosomes attached to it. Main function, we're going to talk about these functions of the ER, the rough and the smooth, in more detail. But the main function of the rough ER is to make proteins. So, what's the function of the ribosome, John? protein synthesis. So if you have the rough ER that has ribosomes bound to it, then the function of that organelle is to make proteins. Smooth ER. Synthesis of lipids, carbohydrates, also detoxifies substances. Right. So I'm going to post the completed cell parts table online. We are going to start talking about cells in general on Wednesday. And yes, so cell differences. So maybe through on page 6, did I get that backwards again? I'm going to post something on Schoology about the reading for Wednesday, where we should be by Wednesday, okay? Awesome. Thank you. You guys get to take any kind of break? You have lab and everything today, right? All bets are off. It's so sad. Oh, you have Dr. Rhodes?